In today's class, we are going to learn about polygenic inheritance. In the earlier class, we discussed about pleiotropy. Both these topics, pleiotropy and polygenic inheritance, are very important from the board examination point of view and also from competition point of view. So let us start with polygenic inheritance. What Mendel has said, Mendel described the discrete differences between the alternative forms of alleles or characters, and it showed discontinued variation. In other words, we can say that Mendel described his characters as tall or dwarf. There was no intermediate form, round or wrinkled. Then we can say purple or white. There was no intermediate form or intermediate alleles in between these two. One, he described them as extreme. One was tall, other was dwarf. There was no intermediate height. So there was no graded variation. But there are some genes which show variation, which show different forms of gradations, like the skin of human beings, human skin, then human height, human intelligence, these show gradation. This means that number of genes, they control these expressions or characters. It is not controlled by one gene, but it is controlled by number of genes. Now these genes are located on the different positions, that is on the different fixed positions on the chromosome, that is which is known as loci. Now human height, human skin color shows graded variation. There are different forms of the skin color. Two extremes are there, that is dark or white, very fair, very fair. Now these are two extremes. One is, if we take height, then one is tall, very tall, you can say giant, and dwarf, very short. But in between these very tall and dwarf, very short, there are graded height. That means there are different variations are seen. Some are not very tall, some are not very short. So you can see different variations even in plants. If we take the example of plants, you can see the size of the seed, the size of the kernel of the wheat, or the color of the fruits. All these show graded variation. The number of seeds are also there. In some fruits, number of seed is more. In some, the number of seed is less. So this also shows graded variation, which are controlled by different types of genes, or you can say controlled by many genes. So you can say that these characters are affected by multiple genes. In animals, we can take the example. In animals, we can take the example of the production of milk in the cattle. The height of the animals, this is also described or expressed by multiple genes, many genes. Because these characters are expressed in the units of length, time, weight. So these are quantitative characters which are continuous. They show continuous variation as compared to discontinuous variation. The inheritance of the quantitative character depends on the number of genes. These genes are independent and they show small and similar effect. The effect of these genes keep on adding to express one character, one trait. The adding of the characters of the gene is seen. So we can say that this is a cumulative effect also. To show one expression or to show one trait, number of genes keep on adding their effects which are small and similar. So this is a cumulative, quantitative and cumulative effect. The traits shown by these genes, that is the multiple genes, they are known as polygenic factors or multiple factors, and the inheritance of the polygenic factors or the polygenes are known as polygenic inheritance. Such genes are described as multiple factors, and the trait influenced by these multiple factors are known as polygenic. So the inheritance which is described by multiple genes or polygenes is known as polygenic inheritance. Now, to understand the polygenic inheritance, let us take the example of human skin color. Human skin color. This is a classic example of polygenic inheritance. It was reported by Davenport in the year 1913. The human skin color is controlled by several pair of genes. So many pair of genes, they control the expression of human skin, and we can see graded variation in human skin color. We all know that the pigment melanin, it decides the color of the human skin. It decides the color of human skin. Now, the amount of melanin present in the skin is determined by several pairs of genes which are located on the different loci of the chromosome. So, we can take a hypothetical example. Say, 3 pair of genes A, B and C. They decide 3 pair of genes. These are 3 pair of genes which decide the color of the skin. This is human skin. So we can say that 6 genes, they decide the color of the human skin. Now dominant pairs we can take A, BB and CC, and recessive pairs these are dominant. The color of the skin, and these are recessive. The color of the skin depends on the number of dominant genes or alleles that are present. We have seen that the intensity of the character, that is the darkness, the more amount of melanin is present if all the alleles are dominant. That means the intensity of the character depends on the number of alleles. So if all the genes are dominant, then the color will be very dark. If one recessive gene is present, then the color is dark. If two recessive gene is present, we can take like this. This is also fairly dark. You can say this is also dark. If three recessive genes are present, small c, then this is intermediate. Intermediate color is C, that is not very dark, neither fair. If four recessive genes are present, then the color is fairly light. Four recessive genes, then this is fairly light. Fairly light means better than intermediate, means little fairer than intermediate or little whiter. Now let's see if five recessive genes are present. This is light. And if six, that means all the genes are recessive. If all the genes are recessive, then the color is very light, means very fair, almost white. So here we have seen, this is a chart which we must remember. So this shows that the amount of the character, the intensity of the character is dependent on the number of dominant alleles. So we see that the number of allelic combination in the gamete is 8 and 64 possible phenotypic combinations can be made when two persons with similar genetic constitution is crossed. Now let us see how is it possible. We take, these are dominant characters and these are recessive characters. Now let us see the example. Parents we can take. The dominant characters are A, capital A, B, C, and the recessive characters B, C. This is dominant and this is recessive. The gametes are these are the gametes, possible gametes. When a cross is made in F1 generation, this is F1 generation. When a cross is made, what we will get is capital A, small a. That means the intermediate color this is intermediate color. So having 3 dominants and 3 recessive. So in F1 generation we get a intermediate color. Now let us see what we will get in F2 generation. Now these are the possible gametes that we get in F2 generation. Now I am not going to draw the Punnett square because it is not asked in any of the examination. It is very time taking. So we must know the amount, the number and the ratio which we get in the Punnett square. That means we are getting 64 out of these 8 gametes, 8 allelic forms. We are getting 64 phenotypic forms. Now these are 8 different allelic forms that are found in the gamete in F2 generation, and a total of 64 phenotypic combinations can be made. So we can get a combination, 64 different types of phenotypic combination when an individual with these gametes are mated. Now these 64 combinations are shown in 7 phenotypic ratios. They are shown in 7 phenotypic ratios. Now these ratios are 1:6:15:15, 20:15:6:1. So we can see the gradation. You see this makes a palindrome. Now you see this ratio, this is very dark. Extreme. You can see the 2 points are at the extreme, and this is very light. This one, 20, it is intermediate. So it makes a bell. When you draw a graph, it will make a bell shape. The curve will be bell shaped. So we have seen the polygenic inheritance by taking the example of human skin color. 3 pairs of genes, they affect the color, that is they express the color of the human skin. So polygenic inheritance means multiple genes act on one character. To show one character, different genes they express. Now in plants, the color of the wheat kernel and the length of the maize cob is also shown by different genes or multiple genes which are known as polygenes. So polygenic inheritance is also seen in plants as well as in animals. In human beings, the color of the skin, the height of the person, individual, the intelligence of a person is also shown by polygenic genes, or it is inherited by polygenes. The production of milk in the cattle is also an example of polygenic inheritance. Now what we see in polygenic inheritance is, it is a quantitative inheritance, means it depends on the number of dominant genes. A character is expressed by the number of dominant genes. Polygenic inheritance is a quantitative and cumulative inheritance. It is based on the number of dominant genes, and it keeps the addition of the genes keep on varying the expression.